I'll be presenting to you the last session of the day. We're going to talk about this very interesting case study. And the reason why we present this case study is because it really gives a nice snapshot of how our platform is useful for three things. Number one, reproducibility, so how easy it is to reproduce work that has already been done, so long as enough of the process is shared with you. Number two, collaboration. So it's very easy to kind of share versions back and forth, to suggest modifications until you get exactly what you're looking for. And number three, which is I think the sort of the more subtle but the most exciting part of the case study, which is that the facility and the user friendliness of the platform really makes it possible to create new and interesting analyses out of data that you already have. So it's very exciting from a research perspective because it's a way of kind of adding more bells and whistles to your analysis, creating benchmarking to actually test out your workflows. Since you're the person who's actually launching your own workflows, now you can benchmark your workflows against each other as well. In order to describe this case study, I'll just give you kind of a background of the story of how we did this project in the first place. We were applying to do a workshop similar to this one in Chile, and another researcher from Chile was also applying to do a very similar workshop at the same event, and we kind of connected over the fact that we both had very similar goals. So Dr. Majacek, whose paper we based this work off of, worked a lot with us. His group did this research on the genetic causes of the Tetralogy of Fallot, which is a congenital heart defect. I'll give you a little bit of background of the kind of the medical science behind it. So we'll talk about what the Tetralogy of Fallot is and what they actually found, but we won't spend too much time on it. So they had already published this study in which they were able to demonstrate that a certain genetic variant was responsible for some cases of Tetralogy of Fallot, of this congenital heart defect. And so Dr. Majacek wanted to give a workshop where he taught his workflow. So really he was interested in teaching people how to do their own bioinformatic workflows. We were obviously interested in something very similar. We wanted to teach people how to do their own work on our online platform. So we kind of combined forces when we realized that we could take his completed work and create a parallel version on our platform to verify his results, which was useful because it created a way for others to be able to go and reproduce his results, but also because it demonstrated how you can take a published workflow and create your own version of it. So in order to do that, we had to find the sort of the equivalent workflow tools from the tools that are available to us that would match up with the work that they did, and that's exactly what we did. From a kind of a high-level philosophical stance, our approach to reproducing the data followed this book by Justin Kitz's, The Practice of Reproducible Research, if you're interested in it. And based on the sort of philosophy that was laid out in that book, we separated the reproduction of this data into two parts. The first part was actually to create a synthetic data set. And this was partly because the data set that was used to do the original study was not very easily available to us. There were different permissions issues, technical issues with how the data was formatted, so it was actually advantageous to be able to create a synthetic cohort, or actually, as you'll see, multiple synthetic cohorts to test against the pipelines. And so in order to do that, we use these two tools that are not GATK tools, but I'll describe sort of briefly what they do, and I'll show you where you can learn more about them. So they are the neat kit and the BAM surgeon, and the way that they work is basically to create a cohort that mimics the statistical distribution of a real cohort, but rearranges all of the data in such a way that it's now no longer associated with any real patients. So you can now do whatever you want to the data because it's no longer associated with any real people. So once we created the synthetic cohort that was supposed to imitate the original, we applied the same variant discovery workflow. We also apply the same or similar prediction of variant effect workflows, and I'll show you where all of these are. We won't actually get to run them all the way through, like you can click launch, but you won't get to the end of where they are. Fortunately, the workspace that you've cloned has already had all of these data sets generated within it. So what we will get to do is actually look at the all the way downstream clustering analysis inside of the notebook that you launched. So the notebook is actually working on data that has already been processed and is already in place for you. So all the computationally heavy stuff has already happened. So the two tools that kind of create the basis for the earlier part of the workflow are this neat kit and the BAM surgeon. So the neat kit is supposed to reproduce the statistical properties of the original data set in simulation. And then the BAM surgeon can spike in whatever mutations you might be expecting. So from our perspective, what was interesting was to make a synthetic cohort that would be similar to the one that the original researchers used, then spike it with the mutation that we expect. So in the sort of hypothetical universe that we've created inside of Terra, the result that they published is the result that is already true. So we have synthetically created a cohort in which a bunch of people have this congenital defect, and they have it because they have this specific variant. And the reason that this ends up being interesting is because, as you'll see, we show that if you do this in two different ways, once you create a synthetic cohort with 100 participants, and once with a cohort that has 500 participants, you get a different result. And so we confirm kind of one of the central positioning aspects of the paper, which is that sample size is crucial to this particular workflow. So these are the first three tools. Actually, this might be a good place to show again where they are inside of the interface. And then, yes, then we can talk about the study itself very quickly. Oh my. Oh, right, when I'm in the view. You know what, I won't switch back and forth between the view because it gets a little buggy. We'll just go to it in a moment. So on this slide, you can see where we matched up the equivalent workflows of what they did with what we have available to us. So for example, they used these pipelines in their paper, which you can see in their preprint, and I'll show you on the next slide where the link to that is in case you're curious. And then these are the equivalents that we use. So we use our GATK haplotype caller in place of whatever it was they were using. And so that's the methodology. You find the equivalent workflow, and you can do whatever you want to reproduce whatever it is that you're interested in proving or in understanding better. So over here you can see links to the preprint and also the final paper. So this is kind of the part that we'll go through a little bit quickly because I don't think everybody is terribly interested in the finer details of tetralogy of Fallot. But very briefly, tetralogy of Fallot is this congenital heart defect. Really, it's 4 separate physical defects. And you can kind of see what they are physically. They have fancy names. So there's an outflow obstruction over here. There is this septal defect, so it's actually a hole that runs between these 2 chambers, and the more your heart has to work to get over this problem, because now it's mixing blood flows, the harder and bigger this hypertrophy over here gets, so that's called right ventricular hypertrophy. So really this is a congenital heart defect that has a very complicated reason for existing. Is it that one of these things is causing the others? Is it that they are all kind of causing each other, which is probably the most true? And from a genetic perspective, what are the genes that are actually associated with these defects? And in fact, this becomes a very complicated question once you consider the difference between syndromic and non-syndromic tetralogy of Fallot. And so in this study, what they were interested in is non-syndromic cases of tetralogy of Fallot, which is to say people who have the congenital heart defect, but not for any reasons that we previously knew that people sometimes have this heart defect. So most cases of tetralogy of Fallot are non-syndromic. This is a problem because, as far as we know, the biggest genetic association between tetralogy of Fallot and another condition would be Down syndrome. But a vast majority of people who have the heart defect do not have Down syndrome. Some of them might have gotten, might have developed rather, the heart defect from some other environmental factors like viral illness or poor nutrition. But how do we tell? How do we tell if we separate out everybody who has tetralogy of Fallot because of already understood genetic reasons, separate out all the people who have tetralogy of Fallot for environmental reasons, and then determine who exactly has it for these heretofore unknown reasons, and then figure out what those reasons are. So that's really what they did, and kind of one of the most important aspects of their study was just the size of the cohort. So they managed to scrounge up almost 1,000 participants, and that's versus 1,200 control patients. So they had these 867 individuals. I believe that's before they tossed out a few, right? So then they wanted to exclude some that had a genetic variant that they were already aware of. And that's really the kind of the premise of their whole study. So now I'm going to switch back over to our user interface. Okay, so I'll start by going into these tools. So real quick, we'll just show you that this is, so this is the entire workflow as I've just described it. The first 3 tools are the ones that create the synthetic data set. So we have one tool that collects a certain, like a template. So it collects this random sampling of people from the 1000 Genomes project. Then the second tool is the NeatKit tool. So you can always click on the tool and see more details. So this is the tool that is supposed to take the real cohort and just rearrange it in such a way that it's now a technically synthetic cohort, but it still has the same statistical properties. Then we mutate the reads with the BAM surgeon, and then after that we run our equivalent pipeline. So we have the haplotype caller, we have joint calling, we have predict variant effects. And so you're very welcome to run these workflows. In the interest of time, we're just going to go directly to the notebook. If you want, you can take a quick look in the data tab. So that's where this data populates after it's been put together. So if you go into the notebook, I'll actually get to show you a couple of extra tricks that I didn't get to show you during my first session. I guess we'll do that at the end. So before we do that, we'll just go through the relevant parts of the notebook. And again, this is a notebook where there's a lot of just kind of clicking through. So if you click all the way down to, sometimes this step takes a little bit longer, and sometimes it doesn't succeed, which is not a huge problem. There we go. It was no problem. How about you? Beautiful. So I like to go through these one by one. It's just a nice way of kind of reading through. So I want to draw your attention to this cell really quickly. This is the cell that actually takes in the test cohort that we created. So that test cohort was populated into your data tab. And so this is the place where we're going to change the code in a moment. So the name of this cohort is A100. That is the 100 participant cohort. So if we continue to click through, these are the various steps that you have to do in advance of the final scoring, which happens right here. So you can see here at the very top of the association is this OBSCN gene, which is not the gene that the researchers were purporting in their paper to be the one that they found with the strongest association. And we were able to basically confirm, as they posited, that it was important to have a large sample size if you want to get a true result. So if we go right back up here and change this to 500, and then we can rerun this cell. Oh, wait for it to finish. And then I'll go ahead and run everything below. And there we go. So there's the difference. So by running the same exact clustering analysis on a cohort that was produced in the exact same way, but just with a slightly higher number of participants, we were able to reproduce the result that was originally published. And this confirmed one of their, maybe not their central claims, but an important claim from the perspective of publication, that having a statistically underpowered analysis is a huge problem, especially because this particular association of this gene variation with this congenital heart defect, this had been noted anecdotally previously, but never proven in a rigorous way. So they kind of purported that, to have proven it by publishing this paper that showed this association because they used this giant sample size, but at the same time, that isn't a proof positive of the fact that they found this association because their sample size was larger. It's a very strong indicator that that might be the reason, but it's an even stronger indicator now that we've performed this analysis. And in fact, if this analysis had been performed before they published their paper, I'm certain that it would have made it into the paper as part of the proof that their methodology was so strong. So actually, this gives us, so good, we have plenty of extra time. This gives us some time to look at a couple of these bells and whistles, and then it looks like we'll be able to take questions a little bit early. I didn't get around to showing everyone, first of all, that you can work inside of a terminal window. So you can click on this button right here, and that takes you to a nice simple bash terminal that is working on the cluster that you've created again. So that's very useful. And sometimes there are certain types of packages that you will have to install through here. It won't work quite as well through the regular interface. Oh, I shouldn't have done that. I should have just clicked close. There we go. And so then I just want to show some more little tricks that you can find in the menu. So first of all, in the file, there's lots of useful versioning tools. You can also download every one of these notebooks in a variety of formats. You can download them as notebooks, obviously. That's kind of the most important functionality, and then in a variety of other types of formats. I really like this edit function. In the edit function, you can find ways to split and merge cells. So I'm going to split this cell right now. The way that the cell splits is it turns whatever is above where your cursor and whatever is below your cursor into two separate cells. I can split those two cells. Now I have two separate cells. I can merge those into a single cell. So if I just click on that and then I say merge to the cell above, now that's back as one cell. We can insert cells. We already talked about running the cells and the different types of kernels, and we've already actually gone through this toolbar up here. So that's really kind of the entire story. I kind of glazed over the details of the Tetralogy of Fallot portion of it, but I'm glad to answer any questions about that if anyone's interested. In the meantime, I hope everyone was stimulated by this example of a case study in which we collaborated with an external scientist in order to reproduce their results that had already been in place. We worked with them, so it was a very collaborative process, and we were able to produce kind of fresh new models out of their data. So that's really what the purpose of this case study as a session is. With that, I would like to thank you all for coming to this workshop. I'll be happy to take any questions. And yeah, that's everything we wanted to show you today.
Are there any questions? Yeah.
Yes. Do you have some website you can point us to with regard to introductory, current, up-to-date versions of how to work in Docker kinds of things?
How to work in...
So I took a Firecloud workshop 2 years ago, and I still have the slides from that, but I was like, let's take something that's more up-to-date.
Well, can I, Addie, we have a question about Dockers, and so is there a place where there's more up-to-date information on Dockers in general, or the ones?
Especially specific to...
Well, you wrote the article, right, in the knowledge base? Is there, there's no articles in the knowledge base.
There are articles, yes, there are articles in the knowledge base on how to use Docker. So if you go right here in the...
I just Googled Docker on like the Terra, learn more about Terra, and there's a whole bunch of like articles written about it that seem just what you're looking for.
So the learn about Terra.
But incidentally, if you don't know, you can click on this hamburger menu and go to Learn About Terra, and it takes you to our Terra support page. We have our we have separate sections in here. So if there are people who are familiar with FireCloud, then you should go to the Getting Started in Terra section. There's a subsection for people who were FireCloud users and just need to convert over to Terra. And then in terms of Docker, let's see, so I believe this is where, yes, so we have a working with Docker section inside of the doing research on Terra section.
So those are pieces of documentation designed to make Docker as easy to use as possible for the average person. Docker itself has a lot more documentation. We were talking earlier about some Windows programs in Docker, so Docker's website and Docker's documentation might be a better source for that level of detail, but we do have some documentation that is up to date for us.
The contact us.
Any other questions?
So while I have this up, I'll also show you that this is where you can go to find announcements, release notes, and also if you ever are in need of contacting us, there are multiple ways to do so. You can click on this nifty Contact Us button and just type out your question. You can go to the community discussion forum and post. And most importantly, we really like to emphasize the request a feature portion of the forum, because so often participants will have such a great question / suggestion, like, why can't I do this? And either we'll have an answer about how easy it is to do it already, or we'll say, oh yeah, that's a great idea. We'll make sure that that button gets made for you. So this is a place where you can submit any type of feature request like that if you want to be getting certain types of metrics, or if there's a, like I said, if there's a certain type of button that you wish, oh, I wish I could just press one thing right now to do this thing rather than tediously having to go back and forth between windows, there's a good chance that we could help you.
Great. Anybody else with questions? Okay, I just wanted to point out this link that I put at the bottom over here in blue for the survey. We're asking that you guys go to broad.io/Terra BroadE survey. And that's BroadE, like the title of the workshop, not BroadE, not IE. Your survey answers really help us to improve these workshops and actually improve the platform too. Our product owners are really interested in hearing what you guys have to say about the platform, whether you like it, whether you don't, what you would need in it, all that sort of stuff. So if you could I think it takes 3 to 4 minutes to fill out. Go ahead and fill that out. I'm also going to be sending you guys an email tomorrow with a bunch of resources in addition to the slides you guys already have, and I'll send the link as well at that point.